I'm going to begin with a little video, but before I do that, just to make a point, because I had a few questions. So what we really focused on in the last lecture was a very small part of what's already a pretty simple system, right? So I talked about how the, I think I have a pointer here, how the canals, remember, so you have the vestibular system, which has 5 organs on either side of your head. We really focused on how the canals operate mechanically as a system, and we came up with this second order differential equation. It has 2 time constants, right, when you solve it, which relates head motion to the motion of the endolymph, right, which is the fluid that fills those canals. And when I introduced the vestibular system, I told you it's involved in generating 2 main types of reflexes. So you have reflexes that drive your eyes, and I showed you a little video with the, somebody walking with a GoPro on their head to show you what it would be like if you didn't have a vestibular ocular reflex. So that's a 3-neuron pathway. So you have basically the hair cells in the canals. You have the afferents, and I talked a bit about how the afferents themselves actually encode motion. Each afferent is only hooked up to hair cells in one of the 5 end organs, so they're pure. They're either horizontal canal afferents or utricle afferents. And then those project onto the motor neurons that move your eyes, right? And then I also talked about this other pathway briefly, and we're going to focus more on that in this, the PPT, that's the 3rd lecture, which are vestibular-only neurons, and they mediate this pathway that's involved in basically maintaining balance, and also send ascending projections up to the thalamus, right? So they're involved in your sense of how you're moving through space. So I put this back up because remember what we did is we went through this sort of mechanical analysis, and we talked about things like viscosities and inertia and elasticity of the cupula. It wants to be upright. So you end up with this sort of mechanically inspired equation to understand how fluid will move through this end organ as you're moving through the world, right? And that was really the goal. And then what we looked at is how do the afferents themselves actually respond? And in that, I should make really clear what you're doing in those experiments, or what the experimenters were doing, is recording from single fibers, right? Single afferent fibers, to understand how individual fibers are encoding motion. And if you look at those different fibers, when you're not moving, so if all of you are sitting in this room, not moving very much, and if we could record from individual fibers coming from your vestibular system into your brain in that 8th nerve, you would find that they're buzzing away at 100 spikes per second when you're just sitting absolutely still. But that discharge, depending on if they hook up to these type 1 or type 2 hair cells, will either be super regular if they're hooked up to the type 2s, or pretty irregular if they're hooked up to the type 1s. So that's individual neurons have different degrees of noise at rest. Okay? So that's just kind of a general conceptual overview. And what I just want to do is show this. So Hopkins was really very proactive around the Olympics, and they put together a little series of videos. I don't know if people have seen any of these, called Mind of the Athlete. So they came to our lab because we work on the vestibular system, and these are 2 of my graduate students. It's Vanessa and Omid. They'd probably be horrified if they knew I put them up on the screen today. But you can find this on YouTube. Hopkins put them out. And what this shows you, so I can talk about how this model that we built of the canals predicts that fluid will move for different frequencies, right? And we had those plots that I showed you, the Bode plots, where you had that low frequency range, mid range, and high frequency range. And we saw the mid range, you had the highest gain, and things were in phase with angular velocity. And I told you at really low frequencies, you can think of the canals as being acceleration sensitive, right? And what happens if you go all the way over to the left on that axis, you're at DC, right? 0 frequency, which is a DC input of velocity. And I showed you for a step input of velocity, you end up with this sort of spike, and then it decays when you actually record from the afferents, and that's what you would expect based on that model that we've built. So what happens in the behavior? You can actually see that at the level of the behavior that people generate. So what I'm going to show you here is, so Vanessa's sitting, you can do this at home with your friends, right? Sitting in an office chair, right? Oops. Oh, I'm not sure what I've just done there. Okay, so Sorry about that. So she's sitting in an office chair in the lab. We've got an eye tracker on her, so we're measuring her VOR, her vestibulo-ocular reflex. That's her eye up on the screen. And Omid's just going to spin her around really fast at a constant velocity, so there's no acceleration, right? It's like a step of velocity. And what you'll see is when she stops, you see this continuation of eye movement, even though there's no velocity. That's because you now have this decrease, right? You have if you look at, so a velocity step that looks like this, you'd have an acceleration here and then a deceleration here. So you can actually see the encoding of acceleration really nicely in this example. So I'll just play a little bit of this movie. There's Vanessa getting spun around. You see her eye generating a VOR. Right? And then she stops, she's staring out into space, and you see that continual flicking of the eye? Did you guys see that? Not really? You can, I won't play it again, but I'll put the link up. So what you see is when she stops moving, her eye keeps moving even though she's not been moving through the world anymore. And that's, again, the property of the canals, because really what they're doing is sensing acceleration at the start of the step of velocity and then deceleration. Okay. Let me just continue where we left off. Okay, so I told you that people have classically looked at the vestibular system with the assumption that it's linear, but that we actually have not really probed until very recently how people actually do move through the world, and we find that you generate really big amplitude movements, much bigger than the linear range that people normally probe, order of magnitude or more larger. In addition, standard linear systems analyses don't account for noise either within trials or across trials. Okay, so then we went through this, and I had a few questions. This is, again, what I mentioned just a moment ago. This is a single afferent recorded in a stationary animal. And you can see each of those little spikes is an action potential. That's a regular afferent. That's the interspike interval. So number of occurrences. Interspike interval is always about 10 milliseconds for this neuron. This is a different neuron. You can see it's much more irregular at rest, so it's an irregular afferent. And these are hooked up preferentially to type 2 hair cells versus type 1 hair cells, right? And then what you can do is just do a Fourier analysis of the resting discharge, and that's what's shown here. Since this is a periodically firing cell at rest, right? You see a big spike at 100 hertz, which is its resting discharge, and then of course you'd see the harmonics. And then for this neuron, because it's quite irregular, you just end up with this kind of relatively flat noise band that basically starts to pollute, you can think of, the area in which we're generally trying to encode stimulation. Because if you did put some sensors on your head to see how you're actually moving through the world, most of the frequency that you're generating is somewhere between 0 and 20 hertz, right? And we know that from experiments that have been done recently. So if you look in this band, there's a lot of room for the actual modulation of the neuron to be sort of untouched by the resting discharge power of the neuron, whereas here you actually have to perturb that resting discharge in order to be heard, right? So you can think of that as an indication of the signal noise that you may have to deal with, okay? So that's just what I was trying to set up there. And then again, what you can do instead of using sine waves, you can use, here we use broadband noise, you can use little snippets of actual motion and see how the neurons, or the afferents in this case, actually encode that. And what I showed you is if you look at a regular afferent, where this is the horizontal head velocity, this is the firing rate, you can just see by eye, right, that they're really close, right? And then instead of using this Gaussian convolution that people typically use to compute the spike density, which was done here, you can actually compute the optimal filter to do the convolution with. And if you do that, you can see you get really good reproduction of the stimulus, right? And then you go do the same thing for the irregular. And then remember, the gain of the irregular afferent is pretty much higher across the whole physiologically relevant frequency range, but it's not as good at predicting or giving you information about what the stimulus actually was, because it's got all that noise. Okay. So you can then go and look at what's called mutual information, which is basically shown here. This is coherence, right? So, and you can see over the frequency range, again, that most of our activity is within. Instead of having higher, irregulars have higher gain, but they have lower information. So what we have now are two channels, one that's really high gain and also has more phase lead, which is the irregular afferent channel, and then another one that actually gives you better patterning of the actual stimulus, right? It tells you really what was happening during that stimulation. So you could maybe think of the irregulars, if you're trying to build something in your mind, as being more like event detectors or transient, indicators. They have very transient phasic responses, whereas these give you more information about what the actual stimulus was. Okay, and then I talked about neural detection thresholds, just kind of, saying that you can use very complicated things to look at people's perceptual thresholds. You can also just put somebody on a chair, and I showed you even a more simple, you can put somebody on an office chair, if you wanted to really, and motorize it. So this is kind of like a motorized office chair. And we know if you do that, we can measure in the darkness that people can make out maybe, a degree per second of rotation, left versus right, maybe 0.5 to 1. That's our limit as humans for rotation. So the question is, how good can a neuron do? And this is what I started to show you guys towards the end. So this is very schematic, and I think this, I appreciate maybe a little confusing. So what this represents is the stimulus, right? So just the head rotation, that's not noisy. We control that, so that's nice clean signal, right? We're just rotating the animal back and forth, or the person if you're looking at behavioral detection thresholds. Then you're going to record the firing rate. This is highly schematic, right? The firing rate is going to have noise on top of it. And maybe the best thing for me to do is now go back over to this curve. So what this shows is the head velocity generated during some frequency of rotation as a function of firing rate for an example regular afferent and an example irregular afferent. So what you can think of to get from neural data to this sort of analysis, first let's just take a cut through 0 this way. So at 0 degrees per second, you're going to end up with some mean of the firing rate, right, for the irregular, which is going to be in the middle of this red band, versus some mean for the regular, right? So they're both going to be pretty close to 100, right? So the mean's not that different. But what you can see is the band is bigger. So if we now go over to here, let's say that this curve represents the distribution of firing rates at 0 degrees per second. If we're talking about an irregular, this band would be more narrow, right? This is the standard deviation of this. And the assumption in this analysis is that the distribution is relatively, normal. It's a Gaussian. And if it was a regular, it'd be broader, right? So that's all. So basically, then what you do in order to figure out what your discriminability is then start with that distribution at 0 and then go progressively either one way or the other, maybe it's asymmetric, until you get two distributions that are further enough apart that when you compute this, you get a d prime of 1, right? So that's what this metric is doing. It's telling you how far apart in terms of velocity do you need to move to get discriminability d prime equals 1. Okay? And of course, if your standard deviation, right, is smaller, you don't need to move as far. So you have smaller neural detection. Okay? So you can do this for regular afferents. You can do this for irregular afferents. What I showed you there was for one frequency. You can do those plots for 0.5 hertz, 5 hertz, 15 hertz, see if it changes as a function of frequency. And generally it's pretty flat for both regular and irregulars, but as you might predict, because irregulars have more noise, they have higher thresholds, right? You need a bigger difference to know if you're moving one way or the other. And then what you can do is compare those right, to human behavioral thresholds, and that's what I show you here, which so of course this work was all done in monkeys because you can't yet record single afferents in people, but presumably it's going to be pretty similar, and monkeys actually have pretty comparable detection thresholds, which behaviorally, which you can measure, you can train them. So here's our regulars, sorry, our irregulars, our regulars. I think this dotted blue line is the best, best regular ever recorded, but you can see here that behavior does much better than single neurons. So it means you need to pool across groups of neurons. So in order to do that properly, what you need to do is know what sort of correlations there are between neurons and other things in order to build logical models of the population. And this actually shows the central neurons that get input, they're even worse than the regulars or irregulars. So here's our central neuron population. It has actually even higher thresholds. So what it suggests that this system is trying to do is pull out something other than just coding of thresholds. It's trying to pull out some other features. Okay. And then I think when I finished, so we'll start on this part, which is, I guess, what we really went through at the very end, is how do we move through the world and what are the implications for coding. And again, with the advent of MEMS sensors, we can actually record six-dimensional movement during everyday activities very easily. And this is important for patients, too. You can actually now start putting these on patients and send them home to do their rehab and see if they're, in fact, doing their exercises the way you want them to do. So this is a very profound development, I think, for like personalized healthcare, as well as understanding some basic science questions. Okay. So I showed you this, and just to make clear what I'm illustrating here, this is a distribution which shows you this. So this is a time series, right? So this would be, say, degrees per second of rotational motion, right? And then this is time, right? So we're just going to focus on one of the 6 axes here. You can do this for the other 5. And then what you can do is plot the distribution of time spent at 0 degrees per second. So normally we're not moving very much, right? So that would be where you'd have the most occurrences, versus these tails that go out to when you're moving really fast, right? So the y-axis here is, well, I guess it's the y-axis rotated 90 degrees, right? So this is basically a distribution centered on velocity, and then of 0 going out to a couple hundred degrees on either side, and then this dimension is the number of occurrences, right? So it's a probability function. Okay. And one thing I mentioned, now this is important for understanding coding, is that if you actually plot in many systems, so here's an example from the visual system, the statistics of the natural world. So people will take pictures of trees and fields and look at the spatial frequency as, and power as a function of frequency, and plot these log-log plots, and what you end up with is a pretty straight line, and says that's following a power law, or things are scale invariant. Basically, you have the same structure at multiple scales. So self-similarity, like a fractal. What we see in the vestibular system is it doesn't seem to have the same form. So this seems to differ from what people traditionally think about both the visual system and the auditory system. I added a caveat that I think people need to actually go back. For example, when you look at visual scenes, you're actually generally not just staring at them, you're making small saccadic eye movements. So as soon as you're looking at any sort of dynamic frequency component, you actually need to consider that too. We're not just passive viewers, we're making even microsaccades when we think we're viewing. So it seems that that may be even more complicated than people think for these other systems. So the point of this is that if you're trying to understand how the system should optimally encode this sort of stimulus, you need to know what its structure is, right? And the idea is that if you want to optimally encode a stimulus, and you're looking at the frequency components, you actually want to sort of whiten the spectrum by the tuning of the neurons. So if you think of the afferents, they actually, remember, have high-pass tuning, right? So by having higher tuning, right, or higher gain at higher frequencies, effectively what you do is multiply this by their tuning, and that actually whitens the spectrum. And this tells you more specifically of what their optimal tuning should be to do that, right? So that's one of the things we learn from doing this type of analysis of natural scenes. We can see whether the tuning of the regulars is better suited to whiteness versus the tuning of the irregulars. And I guess the short answer is it's the tuning of the irregulars, which again is, remember I told you there were two channels, and the irregulars have evolved more recently, right? You don't see them in fish, but you see them in land-dwelling animals. So that makes sense, right? That those are the ones that have now been designed by evolution to best encode this type of stimulus that we generate. Okay. So, so what we've learned is that natural stimuli reach very high intensities, right? They're highly non-Gaussian. I meant to mention that. They don't follow, there was that little curve. Let me just go back. Whoops. Here, this is this would be a Gaussian. Here you can see there's a lot of skewedness. So they have a lot of kurtosis. I mean, you can see that they're kind of long tails, right? So they're not Gaussian at all. They deviate from scale invariance, which was sort of surprising given the literature and sensory systems thus far. You might expect that they would. And the question again is, how do vestibular neurons encode these types of stimuli? Okay. So you can go and measure experimentally. All that work I just showed you is done in people. So if you want to know what's going on, you're usually going to be using an animal model to look at neurons, right? So you might want to check and see what's going on in your animal model first. So you can compare, say, people versus monkeys during natural behaviors. You can record during different sort of normal, everyday monkey and people activities. You can go through the same sort of logic and see whether things are normal, how far are the tails, how big are the velocities we're generating and the accelerations. Are things scale invariant? And what you see is actually natural stimulus statistics in monkeys and humans are actually pretty similar, making the monkey a really good model. You can do the same thing in mice, and now a lot of people work in mice and rats, and it looks like there are some slight differences in mice and rats that might suggest other strategies will be used, in particular coding otolith input. Okay, so let's go back. So I told you we started with linear systems approach, just transfer functions, gain in phase, signals and systems type stuff. So this is your stimulus recorded, say, during with a MEM sensor with somebody running around playing soccer, or a monkey chasing another monkey in the playroom. And what you can do is take that linear transfer function you made and see how well, what should a neuron do, right? So this would be a transfer function made for a neuron when you were keeping within the linear range. And what happens, and you guys can probably guess that, if you've got some low frequency or not so dynamic activity, some kind of mildly dynamic activity versus really active behaviors going on, what you can do is a pretty good job of predicting what a firing rate will be in these first two cases. But as soon as you really push, the behavior, the neurons are going to saturate, right? They only fire at 100 spikes per second at rest, so you're going to cut off in their off direction. You're also going to saturate in their on direction really fast. And that happens very routinely in our everyday activities. So what you need to do is just add, and this is what's done in a lot of sensory systems, basically what's called a linear, nonlinear cascade, right? So what you can do here is have your linear transfer function. Question? No. Have a linear transfer function. You get your linear prediction, but you need some nonlinear function, which is probably going to be like a sigmoid, right? Which will account for that cutoff at low frequencies. Have that linear range in the mid-range, right? If you're looking at basically firing rate versus velocity. So, and then you're going to have this sort of saturation for very high frequencies in the on direction. So that would be probably your nonlinear function. So I'll just show you that. So here's low amplitude movement. Here's the stimulus is in black, right? So little snippets of actual movement that was generated by a monkey. And then what you can do is actually record the afferent and see how it actually responds, and that would be in red, single afferent. You can then make your linear prediction based on really small movements like the ones that people have studied forever. And it does a pretty good match. And then you can put this cascade model where you've also got this sigmoidal function. So this would be your static nonlinearity, where you've got basically at about, things are really only linear over this small range from 0 to maybe 100 spikes per second, right? And here you're actually already cutting off, and here you're cutting off. So by adding this sort of nonlinearity, you do a little better, but you really need that nonlinearity that cascaded into your model in order to predict the neurons firing once you actually expose or apply more dynamic stimuli. Okay, so the summary so far: natural stimuli elicit nonlinear responses from vestibular neurons, right? Because it's very easy to drive them into cutoff or into saturation. Linear and nonlinear models with a static nonlinearity do a pretty good job of explaining what you see if you actually record from these neurons. And one other question is, what about spike timing? So there, you know, I don't know, has anyone talked about spike timing codes yet in this class? A little bit? No? No? Okay. So probably everything you've heard thus far that pertains to neurons has been a rate code. And you probably have a whole lecture on what is the best way to represent firing rate, right? Because really what you're doing is recording binary events, right? Zeros and ones of whether or not a neuron, if a neuron didn't fire, you have an action potentially of zeros, and then in that bin it fired, you give it a 1, right? And then you have to take that basically binary sequence and convert it into something that's more analog, right? Which is a firing rate. So typically what people will do is some sort of, you can do a reciprocal interval where you take, you know, the period and then you take the inverse of that. That's problematic because it's a nonlinear transformation and it's very susceptible to any sort of noise. If you have a false spike, it will really mess up your computation. The other thing people do, which I already alluded to, is a spike density computation where you take a Gaussian function and you convolve it with each action potential and then sum those, and you get a nice kind of smooth representation of firing rate. There you have to determine what the width of the Gaussian you're going to do the convolution is, right? So it has some assumptions. There are other models you can take, Kaiser windows, other types of filters people use. But that's typically, people will, different fields have different preferences, but that's typically the way that people represent neuronal activity, right, is in terms of spikes per second per whatever your stimulus is. Okay. Whoops. So you can look at these two experiments, or two firing rates, and you can see that number of action potentials during a given time window is carrying information. And the firing rates here would be pretty comparable, right? So here, within this same time window, you can see 3 spikes, but they're organized in a very different way. So if you have precise spike timing, here you could have very different things going on within these two conditions, right? So the idea is that here, the basic firing rates look the same over that one window, but here there's something very different going on. I think we're missing a spike there. They should look very similar. But here you can see that there's a different way in which the spikes are organized on a finer scale. So how do you look at that? Here, again, these are irregular and regular afferents. Here's a stimulus. You can see this nice but slightly messier modulation of the irregular. Here the regular is very clean and really reproducing the stimulus. So that's what I already showed you. Then what you can do is, instead of just playing one little snippet of motion, you can actually play a snippet multiple times. Oh, and this just shows the sort of variability. It's another way of showing those plots like I showed you when we did the neural detection thresholds. This is the firing rate as a function of head velocity for one afferent. That's an irregular afferent. And then you can see how things are much more narrow and clean. The gain is lower, right, because the slope is lower here, but the distribution's more narrow, right? So it's just another way of looking at this difference in gain versus noise. Okay, and there you can put where the slope of this gives you a sense of the sensitivity of the neuron, its gain, and then basically this axis gives you a sense of how noisy the neuron is. Okay. So in terms of mutual information, again, regulars have much more information about the stimulus than the irregulars, which I already told you. But now we can look at terms of spike timing codes, right? So we're going to take that same little snippet I showed you and now play it over and over and over again and record from the same afferent for each play of this stimulus. And people can do this. Here I think we use natural stimuli. You can do this for noise. If you do this for noise, it's called frozen noise, right? You just play the same stimulus over and over and over again. And what you can see, I guess we played this maybe, I don't know if it's 7 or so times, that there's this nice banding in the irregular afferent, right? So you get the sense that there's some sort of systematic, I guess, banding or patterning. And here, even though the irregulars have really good information coding, you just don't, it's not that obvious. I mean, you do see something, but it's just not as clean, right? So the irregulars display lower trial-to-trial variability than the regulars do. So how can you quantify that? Well, what you can do is use one of these distance metrics. There are a couple of them. There's, which I'll mention the next slide. So here what we've done is played 3 different snippets, right? And here you record the response over and over again to playing each of these snippets multiple times, okay? And this would be an example irregular afferent, or you can do it for an example regular afferent, okay? And then in order to quantify how different, how much timing is preserved trial by trial for the same stimulus, what you do is you train this classifier. So you say each response to each stimulus is taken, an example response to each stimulus is taken as your template. So you could take the top response to each of those 3 stimuli, right? And then for those remaining responses, you compute the distance between it and the template. And there's a cost function. So there are 2 different ones. So you can go look these up if you're interested. One is called the Van Rossum metric. The other is the Victor Purpura. They pretty much generally will give you very similar results. And the idea is that, if a spike is right on, another spike from the next run, you have very little cost. If you have to move the spike a little bit, you add cost. If a spike is missing, there's another cost. So there are little rules within each of these classifiers. And then based on this cost function, you assign the response to one of the multiple stimuli that you've played, and you see how good the performance is. How good are you at assigning to the correct stimulus? Okay, does that make sense? So if you do an irregular afferent, you pretty much have perfect performance. These neurons are really good at this. And so here's your classifier. A diagonal line is basically performance, original stimulus versus assigned stimulus. You have pretty much perfect performance for irregular afferents. Whereas irregular afferents, you end up with a performance here on average about 0.38. And you're much less likely to have the ability to nail with 100% probability the correct assigned stimulus based on these sorts of metrics. You can do this analysis with different types of windows for the 2 methods. And you can see where you get peak performance. How big a window do you need to do your computation over? And what's really a signature of a spike timing code, so remember I've told you guys multiple times now, if you generate head movements, most of the power that you generate is below 20 hertz, right? So that would be this gray box here. So everything on this side, everything within the gray box is what we're normally trying to encode. And what you can see for the irregular afferents, their best performance is much higher frequency. And so we're up at 10 to the 2 almost. If you look in terms of spike timing precision in milliseconds, it's on the order of about 6 milliseconds for these neurons. Whereas these regular afferents sort of flatten out close to within the high frequency range of normal stimuli. So what this suggests is that you've got something going on at a frequency range or a precision of timing well outside the range of the stimulus you're trying to encode, right? And that's sort of a classic signature of a spike timing code, that the performance of these metrics would peak for a window well outside of the frequency band that you're trying to encode. Okay. So what I've just told you is we have these 2 pathways, right, going in from the 8th nerve. We're still at the 8th nerve. We have these irregular afferents and these regular afferents. The irregulars have big gain, but they have a lot of noise. The regulars have lower gain, much less noisy, better detection thresholds, right, better mutual information. But these guys also have the spike timing code. So what it really suggests is we have two pathways going into the brain, two separate channels of information. So the question is, what happens if you record from the central neurons? Are they preserving one or both of these codes, right? What are they doing? So you can go and do, for example, the same types of spike timing experiments. And what you see, I should just, backtrack. I should note that this was done on those VO neurons. So these are the neurons that are involved in perception, right? We're not looking at the VOR neurons here. We're looking at the perception neurons and spinal cord neurons, not the eye movement VOR neurons. So if you look at the population, so again, these are afferents in terms of firing rate codes, and this would be spike timing codes over here. So you see regulars are more information in firing rate, irregulars less, and VOs less, right? So actually you have less information coding in these central neurons than you do in their regular inputs. And I should note that each central neuron seems to get some weighted input from both regulars and irregulars. And over here what you see is sort of the opposite, right? As far as spike timing codes, the spike timing code is seen in the irregulars and it seems to be preserved in these neurons. So what this suggests, and if you look back at the old electrophysiology literature, is the VOs get a predominant input from the irregulars, even though they get some regular input. So what the prediction is that these neurons actually are interested in transient stimuli, and that the spike timing code is helping convey that. Whereas the VOR neurons, if you do a similar analysis, which remains to be done, you might actually see that they look more like the regulars. And the reason that you'd want them to look more like the regulars is because they have to really generate a very nicely patterned eye movement in order to keep your eye stable as you're moving through the world, right? So their patterning is super important. Okay, so I'm just going to run through the conclusions. The old view, which really, it's funny, I mean, when you see how people actually move through the world, this may seem so obvious to you, but for 50 years, people have been, maybe 40 years, really holding on to the idea that this is a totally linear system. So, the linear models work pretty well in a very restricted range. So you have a linear relationship between stimulus and response, where response is firing rate. The new view, right, is that you have strongly nonlinear responses occurring every day. You know, even if I look from here to here, I've driven my afferents into the nonlinear range. It doesn't take really crazily dynamic movements. Just moving your head will drive them into the nonlinear range. This holds both for rotations and translations. Vestibular coding strategies are constrained by the natural stimulus statistics. So as I mentioned, if you look at the tuning of the irregular afferents as a function of frequency, it's actually very well matched to those scale invariant examples I showed you of the natural statistics of movement, so that you end up with a white spectra. We see temporal coding, which is another word for precise spike timing, in one channel. And that's a nonlinear code, right? And I didn't mention this, but you can also look at things like population synchrony and see if there are indications for any sort of synchrony in the response, which would also be a nonlinearity. Okay. So what can you do with this? This is very interesting from just understanding how the brain represents our motion as we move through the world. And I think it's pretty fascinating if you just think about the fact that you've got these neurons in your eighth nerve telling you in real time how your head is rotating in 3 dimensions and how you're translating through the world. And again, you guys probably we all take this for granted because this system is just working in the background for us 24/7, right? But let's say you have a profound vestibular loss, and I showed you a couple of videos at the beginning of the last lecture. You know, life would be very hard. Just again, keeping your gaze stable, walking, and keeping balance would be very difficult. So currently at Hopkins, there are clinical trials going on for a vestibular prosthetic. So I just thought I'd take a couple minutes to mention that because I think it's very exciting. So the vestibular prosthetic currently is just set up. So people probably all know about cochlear prosthetics, right? You have this basically an electrode with multiple 10 to a dozen sites that run through the scala media of the cochlea. And then you can, take a microphone, take sound, break it into different channels and activate these different sites with certain ratios to reproduce something that's representative of the sound that's being heard. So this is sort of an adaptation of an auditory prosthetic where instead of having one electrode through the scala media, you have multiple electrodes that go into each of the canals. And for the moment, it's really just focused on restoring canal function. That's a bit easier because I remember I told you all the hair cells in each of the canals are oriented with the same polarity, so that their hair cell bundles are all pointing in the same way, with the tallest versus the shortest oriented with the same polarity. So here you just stimulate the ampulla where the hair cells are in, say, the superior horizontal versus posterior canal. And what you can do is put a gyro on the head of a person or a monkey. You can record three-dimensional motion with that gyro. So you have 3 readouts, right, of head motion in, say, horizontal, vertical, and X, Y, and Z rotations, right? And then what you can do is map those movements into the canal planes, right? And what you're then missing is a little box, right? Because what you've got is an analog signal going in, which is head motion in space, and you want to stimulate the hair cells, right? In fact, effectively what you're doing is you're stimulating the afferents. You're bypassing the hair cells. So fortunately, because people do neurophysiology, we know what you might want to put in that box, right? So if you remember, I went through this linear and nonlinear cascade model. That's actually a really good thing to put in this box, right? To map. So you have your head rotation in space. You then project it onto the canal planes. And then to determine how you're going to turn those velocities into a spike train, you put it through that linear, nonlinear model that we came up with to explain how we're going to describe the responses of neurons when you experience head motion, right? And that's currently what's sitting in the prosthetics that, I guess there are currently 4 people on the planet with the Hopkins prosthetic in order to give them a sense of motion. And they actually really appreciate having this prosthetic. They feel that they have a lot better confidence about how they're moving through the world. So I'll just finish this part of the lecture by showing you a little bit about how that prosthetic looks. So here's the gyroscope that sits in, it's on an external magnet. So you have basically a magnet under the skin, and you can kind of then mount this to the magnet. It has a microprocessor that converts head velocity into a pulse rate using, again, this mapping function that's based on real neurophysiology. So it's a nice place where basic science informs prosthetic design. The electrode arrays are implanted deep within the canals, okay, and can stimulate the vestibular nerve. Okay, so there's where the canals are. And then the vestibular nerve is carrying the information into the brain. Okay, and as I mentioned, you can take these sorts of, this would be that static nonlinearity I told you about, right, which is basically a sigmoid, and use that as part of the mapping where you have the linear range, which you'd have your transfer function, right, that you estimate over that really restricted area. And then you also have to add in this sort of static nonlinearity in order to really map head motion into something that these patients can use. Okay, so just in summary, what I've told you, I think I've said this a couple times now, so I'll go through this quickly. Regular afferents use conventional changes in firing rate or rate coding, and that's probably important for VOR pathways. So if somebody went and looked at the PVP neurons, the position vestibular pause neurons that are the direct neuron in that 3-neuron arc that generates the VOR, afferent, central neuron, muscle, motor neuron that moves the eye muscles. They may look more like the regular afferents, to be determined. And irregular afferents are highly nonlinear and transmit information using precise spike timing, right? Well, I say 5 to 6 millisecond precision. And they're involved in vestibular spinal pathways and ascending pathways. And I think I already made this note about the evolutionary perspective, so I'll stop there for that. Okay. So now I'm just going to move to another topic. So the way I just described, okay. So what I focused on here in the last lecture was, sensory coding in the vestibular system. Okay, and I said we talked about what are natural movements like. We actually played natural movements in. We got came up with nice models that we can use to stimulate a prosthetic. And that's the kind of approach I went through is a really very engineering, very engineering-inspired approach to sensory neuroscience, right? But if you think about the way you activate your vestibular system in everyday life, it's generally not sitting on one of our hexapods being, stimulated. The way when we're recording from our neurons, we're actually, stimulating these animals by applying certain types of stimuli, right? Like the frozen noise type experiment where you play an epic of a stimulus over and over and over again is somewhat artificial, right? It tells you that there's a code there, but the stimulus is applied. And in everyday life, generally you're activating your own vestibular system, right? Unless you have a really long commute. Most of the time, you get up, you sit down, you move. Even if you're commuting, you're moving your head back and forth. Those are all voluntary movements, right? And if you think about it, it's really important that you're able to dissociate sensory input that comes based on things you've done yourself versus unexpected things, right? So has anyone here ever experienced an earthquake? Yeah. So I was in Chicago as a graduate student. They don't have a lot of earthquakes there, but they had a little tiny earthquake, which is a very salient event, right? Like when the world moves, when you don't expect it to move, and you're like, whoa, I didn't do that, right? And it doesn't need to be a lot of movement. It can be like just a tiny bit of movement, but it's a very salient event. So the brain somehow figures out very quickly that I didn't do that. So most of you, if you've experienced an earthquake, you probably went through that. It's like, wait, I didn't do that, right? What's that, right? It's a very profound and probably important ability, right? Okay, so we need that for perceptual stability. Hey, I didn't do that. Something happened. And it's been long appreciated that this is important. And recent work has focused on understanding the neural encoding of vestibular information that distinguishes active self-generated motion versus passively applied movement. And basically the terms that are used in the literature, if it's externally applied, it's X-afference, so that's easy to remember. And then if it's something that is caused by something you've done yourself, it's called reafferent. So you have two types of sensory input that you can experience, right? If I shake my head back and forth, that's reafferent vestibular stimulation. If somebody moves my head back and forth, that's X-afferent. Okay, so how does the brain encode passive self-motion? There are two types of channels. I already told you about that, so this lecture, okay. And again, most of what we do is active. Okay. So the first question is why. I told you the idea that you need to know if you're experiencing an earthquake or if you're, just moved over to tie your shoe. And Helmholtz back in 1867 made this point, so you guys can try this too. You can tap the side of your eyelid, don't tap your eye, right? And the world's going to jump, right? You get that little jump. But if you make a saccade, the world is stable, right? Even though your eye is moving probably faster if you make a saccade from here to here. And why is that? Well, in one case we have this gaze command our brain is generating that it can keep track of, so it can fill things in and keep the world stable. The other condition's pretty not natural, so the brain hasn't really evolved to deal with you tapping the side of your eye and keeping that stable. So you need that for perceptual stability. The world stays stable, right, during saccadic eye movements. You also need this for accurate motor control. So consider the example of the vestibular system. The same neurons that get direct afferent input project down to centers that make vestibular ocular and vestibular spinal reflexes. And particularly the vestibular spinal reflexes, if I'm walking on ice and I slip, I want my postural responses to keep me upright. If I move my head the same way because I want to return a tennis serve, I don't want to be mounting these compensatory reflexes to keep me upright, right? I want to return that serve. So when you're actually generating involuntary movement, this 3-neuron arc that generates vestibular spinal pathways is counterproductive, right? So you might actually want some way to modulate that based on what your intent was. So here's just a really clear example. I think there's an ice storm in Montreal today, which made me glad I moved to Baltimore. So this would be somebody walking down the hill from where my lab used to be, slipping on black ice. Hopefully, they've got a robust vestibular spinal reflex, and they're going to catch themselves, right? Okay, that's X afferents, unexpected movement. And then you have somebody doing some complex sport routine, maybe moving the same way through space, so the vestibular system has the same input, but she actually wants to complete her jump, right? She doesn't want to be stopped, right? So here you want the vestibular spinal pathways in a very different state, and this would be self-generated reafferents. So I already told you about these two pathways, so here we're going to focus on this pathway here, the vestibular spinal pathway, and record again from afferents initially, because there is a pathway by which the brain could actually change the way that the end organ is sending information. It's called the efferent system. Did you guys learn about the auditory efferent system at all? No? So you always think of sensory information just coming in from the ear, but in both the vestibular and the auditory systems, you actually have efferent cell bodies that sit in the brainstem that send information back out to the ear. And that's both the case for the auditory system and for the vestibular system. We're still trying to understand what it does in the vestibular system. There's some clues in the auditory system. It's involved in the cochlear amplifier and tuning the cochlear amplifier. So the reigning hypothesis had been that during active movements, you send an input, something in from the brain that says, you know what, you're going to make an active movement, maybe raise the resting discharge a little bit so you don't cut out, and decrease your gain, right? So that was the idea. But you can do that experiment, and if you record from, these are passive head and space movements generated by rotating an animal that's head fixed back and forth, so you're just stimulating the vestibular system. And these are active movements that the monkey generates himself. You can actually take the movements the monkey generates and play them back into the turntable so they're perfectly matched. And you see that the neurons respond the same way. Okay? So you can fit this with a very simple function with a resting discharge, a head velocity, and a little bit of a lead term, which is here an acceleration term. So again, looks like our basic second order differential equation. And I'll just stop here because I realize I'm running a little late. So the idea is that if you compare passive versus active movements, there's no change in the sensitivity or the gain of the neurons, and they're the same in both conditions, and their discharge bias doesn't change. So there's no change. So what I'll do for the next lecture, and I'll also talk a little bit about cerebellar processing, is then move to the next stage of processing and tell you what happens in the central pathways at the first central stage of processing.